Biogas. What effect does pH have in biogas production?
Alina Hassan.
And Sara Abdurrahim.
Introduction. Carbon emissions have been a huge dilemma in today's society. Global warming and climate change are two words used frequently when describing the outcome of too much carbon in the atmosphere. Climate change is when the weather over time slowly changes. Global warming is when the temperature warms over time or the earth starts to become hotter over time. Pollution is another term to describe this matter. Pollution is the term describing when toxins are released into nature. In this case, the toxin is carbon. Currently, only 1% of Earth cannot inhabit life, but by 2050, it would potentially rise up to 20% due to climate change and global warming. Climate change is also endangering many species, like polar bears, ringed seals, koala bears, monarch butterflies, etc. Many people are struggling to deal with this effect on their habitats and the drastic changes of weather. Electricity and heating through burning fossil fuels pay a big contribution to climate change. For our project, we are going to do biogas production. We want to inform consumers about the best way possible to produce biogas. Biogas has been explained as an amazing alternative to many major issues having to do with fossil fuels. We wanted to know if the pH of biogas will affect its production so we can understand if it speeds up its production and works more efficiently.
Our big question is, does the pH of our biogas affect its production rate, or how long it produces heat for?
Abstract. What we chose to do for our project is to figure out whether the pH of biogas affects its production. The way we conducted this experiment is by creating two anaerobic digesters, both having different pH levels, and tested to see which one held the flame for the longest. By performing this test, we will learn the difference between biogas with a higher pH level and one with the standard pH level, allowing us to figure out the preferred pH level of biogas to create a more efficient and productive version.
The purpose for our project is to figure out if biogas production is more effective with a higher acidic factor, to comprehend if the pH of biogas jeopardizes or helps with its production. We wanted to know this because consumers wanting a more sustainable alternative to the burning of fossil fuels could know the most effective way to create heat. Biogas can also be used for many purposes, but we are demonstrating it from the aspect of heat production.
This project contains a fire hazard and should not be done without adult supervision. We advise you do not attempt this experiment in the manner that we approached it, whereas it was very unsafe.
What is biogas? Biogas is a renewable energy source, natural and beneficial towards the environment. Biogas consists of broken down organic matter such as food waste, compost, and animal waste. It is broken down by microorganisms through this process called anaerobic digestion. It can be utilized for electricity, heating, and fuel for cars.
The benefits of biogas. Biogas is eco-friendly. Biogas assists with reducing the dependency on fossil fuels such as coal and oil. Biogas is also a much cleaner, renewable, and sustainable source of energy. Cheaper. Technology used to produce and generate biogas is significantly cheaper, much simpler, and accessible considering that the things such as food, waste, and animal waste can be used to produce it. Cleaner planet. Biogas production may improve the quality of water and reduce waterborne diseases due to the way it's produced through anaerobic digestion. Anaerobic digestion could help deactivate harmful parasites and pathogens, organisms that cause diseases to their host. This can improve the water quality, environment, and the overall cleanliness of the earth. Creates natural alternatives. Through the generation of biogas, anaerobic digestion, organic digestate is created, which is a wonderful and healthier substitute for chemical digestate.
Disadvantages of biogas. Similar to other renewable energy sources, biogas is affected by the temperature and weather. The optimal temperature that is required for bacteria to digest is around 37 degrees Celsius. This makes it difficult for biogas to become our main source of energy due to the constant shifts in temperature. An unfortunate disadvantage of biogas production is the fact that there isn't any new technology that has been created to produce large-scale amounts of biogas for larger populations. This makes biogas far more inaccessible and expensive, and unfortunately, many governments aren't willing to invest into this technology. Biogas also does contain impurities. Even after refinement and compression, biogas is still likely to contain impurities, which would corrode the metal parts in a car if it was used for one. This would obviously lead to many issues and maintenance expenses. This is why biogas is better when used for smaller objects such as lamps, light bulbs, and water boilers. Biogas is also not as accessible for many. Industrial biogas production is only available when the amounts of materials required, such as food waste and manure, are plentiful in supply. This is why biogas generation is better suited for rural areas with lower population.
How is natural gas and biogas different? Many people think that natural gas and biogas are the same concept, when in reality, they differ greatly from each other. The biggest difference when it comes to the two sources of energy is that they are both perceived as renewable. Natural gas is not a renewable energy source and consists of mainly methane, CH4, which isn't renewable, as biogas contains a minimum amount, leading to less of an impact. Additionally, something that baffles many is that natural gas isn't as helpful towards the environment as it seems to be. Biogas helps lessen carbon emissions, while natural gas seems to slightly help it grow, due to its methane content. It still allows CO2 emissions to be reduced and still helps the environment. How does it contribute to carbon emissions? When the methane is burnt, it releases CO2 into the atmosphere, leaving a remaining ecological footprint along the way, and it can be impactful over time. Furthermore, natural gas has an extensive ability while biogas has a restricted supply and is not so widespread. Resources are hard to produce in certain circumstances.
Biomass versus biogas. Biogas and biomass are also often seen as the same thing, as most individuals are quite confused about the differences between them. Biomass is in the form of a solid, and biogas is transparent and in the gaseous condition. Biomass utilizes organic substances and burns it in order to produce energy, whereas biogas goes through the anaerobic digestion process in order to give out energy.
How does the anaerobic digestion process work? The anaerobic digestion process has four phases: hydrolysis, acetogenesis, and methanogenesis. First, in hydrolysis, specific bacteria breaks down organic polymers and converts them into simple sugars. Second, in acetogenesis, a specific category of bacteria called acetogenic bacteria turns simple sugars and amino acids into hydrogen, CO2 organic acids, and ammonia. Third, in acetogenesis, during the stage of acetogenesis, a particular bacteria named acetogenic bacteria turns the organic acids into hydrogen, CO2, and acetic acid. Fourth, in methanogenesis, specific singular cell named methanogens change the intermediate products created in the preceding processes into biogas, mainly methane and carbon dioxide. The digestate, solid and liquid product of this process contains substances that microorganisms cannot consume, as well as deceased bacteria.
What is an anaerobic digester? A huge variable playing a role in our project is how we can produce the biogas. Anaerobic digesters are the very thing used for biogas production. Anaerobic digesters are tanks that are deprived of all oxygen, and it performs a process called anaerobic digestion to create the biogas. Its main purpose is to transform organic matter into biogas and digestate, which is a remainder of waste after the anaerobic digestion process.
pH. One of the biggest points we have to cover is what pH is. The pH of fluid solutions is an objective indicator of their acidity or basicity. Acidity refers to the attribute to anything acidic, a chemical element, usually a liquid, that includes hydrogen and can combine with other chemicals to generate salts. Basicity is the condition of being a base, a substance that can counteract the acid by responding with hydrogen ions. In our project, we need to use a pH strip to observe our biogas's pH levels. You may be wondering, what is a pH strip? A pH strip is a strip of litmus paper used to determine the pH of a liquid. The material on the paper makes the strips represent different colors at different levels of acidity. There is a scale from 0 to 14 to determine the different pH levels called the pH scale. On the scale, 0 is quite acidic, and 14 being quite alkaline. Alkalinity is a measure of a water's body's ability to balance the acids and bases, thereby keeping a relatively steady pH level.
What is a pH buffer? pH buffers are solutions that prevent large alterations in pH levels when an acid or base is added to it. It creates a stable pH for a solution by helping with maintaining it in a specific range. Buffer solutions are essential in various biological, chemical, and environmental processes which require a stable pH level. Buffers don't only balance pH levels but can additionally change the pH level. Vinegar can act as a buffer since it's a weak acid and its ionization is less than 10%. Another term that's more fitting is an acidic pH adjuster. But since we are combining it with water when we add it into our anaerobic digester, the pH lessens, making the term pH buffer to be a better description.
How does biogas differentiate from other renewable energy sources? Biogas has a few differences when it gets compared to its fellow renewable energy sources. Initially, the most noticeable difference is that biogas derives from organic waste, whereas other renewable energy sources utilize light from the sun, solar energy, wind energy, heat from the depths of the earth, geothermal energy, etc. Also, biogas utilizing hydrogen has less carbon strength than another well-known fossil fuel called solar energy, which uses the sun to produce energy, as I said previously. Biogas is as well categorized as a fossil fuel without the negative ecological impact, unlike the majority of renewable energy sources. Also, biogas has more availability than some other renewable energy sources. Biogas can be produced at any time you desire, whereas solar energy and wind energy, it must be produced when there is enough sunlight or wind. How does it reduce greenhouse emissions? Methane capture. Methane is a powerful greenhouse gas with far higher global warming potential than carbon dioxide. It's responsible for over 25% of the global warming we are experiencing today. By seizing and utilizing methane produced during the anaerobic digestion of organic waste, biogas systems help with preventing the release of methane into the atmosphere. Replaces fossil fuels. Biogas can be a substitute for the traditional fossil fuels that we use today. By replacing the fossil fuels with biogas, we will be able to decrease the overall carbon footprint, and it reduces the reliance on non-renewable sources of energy. Returning carbon to the soil. Through the process of anaerobic digestion, you are left behind with a rich nutrient slurry, which can be utilized as fertilizer. When this fertilizer is used in agriculture land, is restored. The carbon is restored back into the soil, enhancing the soil's health. This helps by reducing the greenhouse gas emissions by promoting carbon storage in soil. Waste management. By properly and effectively managing organic waste through biogas production, methane release is lessened, resulting in overall reduction in greenhouse gas emissions.
Hypothesis for our project is if there is a comparison between biogas without an added pH buffer and biogas with an added pH buffer, then biogas with the added pH buffer should produce the most heat because its production rate would be faster due to its acidity.
The materials required for our experiment will be plastic tubing, vinegar, drill, pH strips, compost, lighter, strong tape, lemon zest, which is optional to speed up the process, 4 glass jars with lids, silicone tubing, infusion set, and 2 of them, grater, syringe, labels, stopwatch, or foam stopwatch. Procedure for the anaerobic digesters. First, gather compost or organic waste. Our choice was food scraps that you can find at home. Also add lemon zest, which you should shave off the skin with a grater. Second step is grab 2 mason jars. Third step is drill a hole in both of the lids of the mason jars. Fourth step is put water into one of the jars. We will call this jar number 1, which shouldn't be hot or shouldn't be too cold. It should just be right in the middle. In the other jar, we should put around 1.5 cups of compost inside of it and add around 187 milliliters of water into the jar. We will call this jar number 2. In jar number 1, drill another hole to the left of the lid. Seal the lid tightly onto the jars. Remember that jar number 1 has 2 holes. And tape around the lid seal so it won't open, allowing no air to escape. Eighth step is to grab the silicone tubing and cut it in half. And the rest we will use for the second anaerobic digester. 9. Place one end of the silicone in the first holes that we made in jar number 1 and jar number 2, the ones in the middle of the lids. 10. Cut the bottom part of the IV set off and make it as close or as far as you'd like, and the bottom part is the syringe with the cone at the end. 11. Place the new end of the IV set onto the other hole in jar number 1. Add a label into one jar saying no pH. Repeat for the other anaerobic digester, but this time add 80 milliliters of vinegar to the jar with organic waste and label this one pH.
Procedure for the testing. As we mentioned earlier, we advise you to order an IV set with a needle, and the steps on the slides that are colored in red will be the steps we did but were not advisable and what we used instead of a needle. So step 1 is to grab your anaerobic digester. Step 2, grab the end of the IV set. Step 3, which is also not advised, so yeah, place around 5 centimeters of extra tubing onto the mouth of the IV set. Make sure the holes both align. Step 4, tape them together. Once again, make sure that the holes are aligned. Step 5, turn on the lighter and set the new mouth of the IV set ablaze. You can just light up the needle on the IV set on the step if you have a needle. And step 6 is to use a stopwatch and observe how long it remains on fire for.
pH of our anaerobic digesters. We tested the pH of our anaerobic digesters to represent the different levels of acidity. It might not be noticeable, but the anaerobic digesters' pHs were slightly different. The pH levels of the anaerobic digester without the pH buffer was a 6, and the anaerobic digester with the pH buffer was a 5, showing that the anaerobic digester with the pH buffer is more acidic.
Data analysis. First, we wanted to quickly go over how the experiment went and some thoughts we had on it. We initially thought that the results were just from the plastic burning and we were worried, but when we went a step further and tested the remainder of the extra tubing after we were done, and the plastic held the flame barely and instantly burnt out in less than 1 second without the lighter. Moreover, the flame with the anaerobic digester was really high when it was put with the lighter, and the extra tubing alone didn't achieve the height as with the anaerobic digesters. For our first attempt on the anaerobic digester with no pH buffer, held the flame for 4.15 seconds. The second attempt held it for 3.26 seconds, and the third held it for 5.37 seconds. The average was 4.26 seconds. We calculated this for comparison reasons. For our first attempt on the anaerobic digester with no pH buffer, it held the flame for 3.21 seconds. The second attempt held it for 3.04 seconds, and the third held it for 2.51 seconds. The average was 2.92 seconds. The anaerobic digester with no pH buffer noticeably held the flame for a longer span of time. This was very shocking towards us as we anticipated the anaerobic digester with the pH buffer to hold the flame the longest.
Sources of error. There were various potential sources of error that we would need to address. One of the potential sources of error that we should have replaced the water with the vinegar, which is the pH buffer. I think that since there was more moisture in the anaerobic digester, or the vinegar became more diluted, it might have affected the outcome, or manipulated the pH. Additionally, we could have used an IV set with a needle. We initially thought that the IV sets were accompanied with a needle, but we were mistaken. That might have slightly affected our results, whereas we used extra plastic tubing as a replacement, and that was dangerous. Furthermore, we had the anaerobic digesters go through the digestion process for only 4 days. Usually, people do at least a week, but we thought that since there was less space, and since we were just making smaller anaerobic digesters, although it might have affected our end results, we made them shorter.
Conclusion, our hypothesis was proven wrong. We expected the anaerobic digester with the pH buffer to hold the flame for longer than the anaerobic digester with the pH buffer. After conducting the experiment and performing the test, we were left with the result that biogas that hasn't had pH buffer added holds a flame for a longer period of time. The results for our multiple trials at testing the biogas had all left us with similar outcomes. The biogas without a pH buffer is superior. This experiment gives us an idea on how to assist with the production of biogas by figuring out the better average pH level for biogas generation, which is an important variable in its production. Throughout the experiment, we came across various challenges and difficulties such as missing materials and having to substitute certain things, which led to attempting many trials. Our results are a crucial part of the future of biogas. With our results, we know the most efficient way to produce biogas, which can help turn biogas into a more accessible and well-known resource.